Okay, good afternoon. Welcome to the Hempcelium presentation by the M2Bio Sustainability Division. This afternoon, I'll be presenting with Aidan Price, our genetics researcher and scientist. We've been working on the project for a little over a month now, and very excited to share with you the future potential of what we've been researching. So without further ado, I'll introduce you to Aidan Price. He's completed his undergraduate BSc in molecular biology at and biotechnology at the University of Stellenbosch in 2019, and then at the end of last year completed his honors degree in genetics, also at the University of Stellenbosch. He's currently performing research for the Molecular Breeding and Biodiversity Lab, where he does conservation work focusing on identification of shark and ray traded products, while also performing population genetic assessment on threatened species in the Southwest Indian Ocean. Aidan has co-authored an article under review for the African Journal of Marine Science relating to his work. I'll let you tell him more about his interests.
Thank you very much, Luke. I think you pretty much summed it up. Most of my work has been involved in conservation genetics with sharks and rays, although I do have many interests which lie outside of that, one being Mycelium composite materials. Once I found out that M2Bio was involved with the Mycelium composite materials, I contacted Luke, obviously, and we had lots of interesting conversations. And now I'm mostly interested in the optimization of fungal strains for the Mycelium composite materials. I came to realize that there wasn't much of a conversation about the genetics and the biology behind the actual fungal strains used. So I'm quite interested in optimizing those fungal strains to try and produce the best possible material that we can. I'll probably delve in a little bit more of that a little bit later. But yeah, that's me.
Thanks, Aidan. Yeah, just to say that we obviously work well together now because I'm the mechanical engineer focusing on the mechanical characteristics and those properties, and Aidan comes with his experience of genetics.
So today, just a little brief introduction of what we'll be doing. We'll be going through the general workflow, how we go about what we do, the production process, and everything behind that. Luke will give an update on the surfboard, and then I'll dive into some of the genetics and future potential things that we could look towards and some exciting prospects. And then just to wrap it up, we'll talk about the need for a packaging alternative.
Great. So just to give you a reminder of our workflow, and then this will also give you an overview of what we'll be discussing in a bit more detail further on. So in the first stage, we get our substrate, our strains rather, of Mycelium, which we then put into a sterilized substrate or grain spawn that's been either pasteurized or sterilized in order to kill any bacteria or other contaminants. We then mix the sterilized substrate with the mycelium and then add it to the mold so that the mycelium can grow through the substrate in the mold, removing it from the mold to allow the aerial hyphae to grow for a short while before baking it and creating the preferred surface we want for our mushroom packaging products. Okay, so into a little bit more depth in each of those steps, starting at the beginning with the Mycelium syringes.
So as I said briefly, I'm kind of interested in the fungal strains used and seeing if you can optimize that. First thing I thought was, obviously there's many different species, many different strains that you can use. Many have been tried with Mycelium composite materials, ranging from reishi, turkey tail, and obviously we've ended up on oyster mushrooms, which we found to be the most effective, and it's been proven effective through multiple sources in the use of Mycelium composite materials. Not only that, but it's tenacious and robust, and it has all the desirable growth characteristics we're looking for in a fungi. And also the final product integrity shows something that we're looking for, something lightweight and mimics a foam, which is what we're looking to currently replace currently used packaging materials such as expanded polystyrene, polyurethane. So we're trying to find something that will give the final product integrity in comparison to that.
Cool. So after the strains, we're obviously interested in the substrate we're using. We're using hemp, and so the discussion is around pulverizing the hemp.
Yeah, and so there's a discussion, as we just said, between the pulverized and the coarse substrate composition. Both have pros and cons. Right now, we have a lot of pulverized hemp stalks that we're using. The benefits of this is increased surface area, which will benefit growth. Other than that, the denser substrate will make it harder for the mycelium to grow throughout the material. Not only that, but an increase in heat and carbon dioxide isn't good for growth. In comparison, the coarser substrates will lead to better aeration, better oxygen availability for the mycelium to grow. And with the coarser material, there'll be more air pockets in the substrate, obviously for the supply of oxygen to the mycelium. So right now, we're in a conversation between pulverizing coarse and trying to find a decent ratio to use.
These are the two broader varieties that we can get it in. So there's a machine that can pulverize it finely or coarsely, and we've been working with them separately, but we're looking at experimenting with them in different ratios for the optimum growing medium. Okay, then this is how it all begins. We modify our console jars to have silicone self-healing ports. We sterilize the grain, which is first soaked and boiled for a period. We put it into the jars, sterilize it within the pressure cooker, and then inoculate it with the syringes. As you can see on the left, we can also add substances such as gypsum and coffee grounds. It's a good opportunity to upcycle the coffee grounds and reduce food waste. This improves the growth of the mycelium. And as you can see from the images on the left, before inoculation and then after 2 weeks after inoculation, the growth of the mycelium. This is also important for scaling and colonizing, taking from a single syringe and making a lot more mycelium in order to use for the packaging. And then we're going to be doing the sterilization or pasteurizing of the bulk substrate. So taking from the grain spawn, you then add it to the bulk substrate before putting it into the mold. So this is all about scaling. And one of the best methods that we know of is lime pasteurization, which we've now tested, and it's proven to work. It's worked across. It's a proven method that's been used before. It also, lime is good for creating hempcrete, making it dual purpose in its purchase and saving us cost potentially if we're doing other experiments with hempcrete. Another type of pasteurization is the hydrogen peroxide, which is a natural substance. It does, in high concentration, it can be quite dangerous, but in low concentrations, we use it to sterilize the substrate. It also partially dissolves the hemp, making it more bioavailable to the mushrooms. And the mushrooms actually do then decompose it and turn it into water and oxygen, which they can also use as they grow through the substrate. Then the final one, I'll let Aidan explain.
Yeah, so this is another type of sterilization that has been interesting to look at. The saltwater solution, or just a water solution. And the basic principle behind that would be soaking the substrate in the solution for up to 2 weeks, which will lead to the growth of anaerobic bacteria. And after 2 weeks of soaking the substrate, you drain the water, allowing for the substrate to air dry, leading to the killing of anaerobic microbes where oxygen essentially acts as the sterilizer. This is obviously a very interesting way, a different way to sterilize the substrate, a lot cheaper, and water is obviously a lot more accessible. The only problem is the longer lead time. This is something that we haven't experimented yet, but we're looking forward to trying it with our hemp and mycelium strains in the near future.
Yes, I should have emphasized that with the previous two. We generally take 12 to 24 hours pasteurizing with these two methods, as opposed to the salt water, which can take up to 2 weeks. But of course, the salt water is readily available to us, and so we look forward to experimenting with it.
And then obviously the final product, the substrate is then inoculated with mycelium and placed in a growth mold. Or sometimes we do put in a pre-growth container to allow the mycelium to grow for a bit before we physically break up the mycelium, just to ensure a homogeneous distribution of the mycelium in the substrate. And then upon the completion of growth, we remove it from the mold and then allow it to grow for a few extra days for the growth of aerial mycelium, which is mycelium which grows at the substrate air interface. And that will just allow for a foamier exterior and more desirable aesthetic and mechanical properties. Once that is done, you dry it at 60 degrees Celsius to kind of solidify the product and also to heat kill the fungi to prevent further growth of it. throughout the substrate, and then that is the final product.
Great. So now that we've gone through the whole process, let me remind you about the Surfboard Project and how it relates. So surfboards are traditionally made majority out of petrochemical products, from the polyurethane EPS core to the resin and fiberglass outer layer. There's petrochemical products throughout. So we decided to make as much as we could, fully renewable surfboard, where we can biodegradable as well, in order to prove that many of the materials used in surfboards are also and translate into packaging. So this is sort of a vector for our project. The full PLA frame is now completed, as you can see below. The image on your right is made on a CAD software. And after quite a lot of experiment with the 3D printer, we managed to fully print the surfboard, and it is now in one piece in the labs in Hop Bay. We're then going to coat the surfboard in a hemp cloth and bioresin, making it as renewable as a performance surfboard gets at the moment. So this is the third iteration of the design. We originally had a honeycomb structure, which we found to be too heavy. And before that, we had this much more hexagonal with smaller shapes, but this just didn't work on the 3D printers. You can see the finish is very poor. So finally, we settled on our design and look forward to colonizing it with the Hempcelium. And yes, that will be happening before the end of this month. So yes, looking forward to handing over to Aidan to tell us more about the genetics and experiments we're going to be doing there.
Thanks, Luke. As I've said before, mentioning fungal strains over and over, obviously it's an essential component of the Mycelium composite materials. And with the advent of multiple biotechnologies using living organisms for production processes, there's a lot of optimization that goes into these living organisms so as to get them to perform in the most efficient way possible. And right now, there are several molecular and genetic techniques that can be used to optimize fungal strains for mycelium packaging. With the three below headings here, kind of the areas which we'll focus on. One being the mycelium growth rate, one of the most important factors, obviously. Right now, it takes several days to fully grow the mycelium composite materials. So improving growth rate would be a huge increase. Mycelium density, which mostly affects the mechanical properties of the final product. It's important to have a lightweight product, but a dense enough material so as to have these mechanical properties that we desire and to meet certain standards that we are looking for. Also to be considered as an alternative to currently used packaging materials. Those are the mechanical properties that we're looking for. And lastly, homogenous growth. As we said previously, sometimes we'll grow the mycelium substrate in a pre-growth container, shake it, and then just to ensure homogeneous growth. But sometimes growth won't be as homogeneous as we like. Sometimes it doesn't penetrate well through the substrate. Sometimes it just grows at the surface. So that's also another factor that we have to consider when optimizing these strains.
These are all the things we're going to try influence in order to compete with the commercially available products at the moment.
And so breeding is one thing that I've thought of. Breeding is an efficient way to generate new fungal strains with improved and desirable traits. I'm currently planning a breeding program with selective breeding, breeding two individuals with each other, where each show desirable characteristics. I'm planning on acquiring a wide variety of oyster mushrooms, trying to get genetically like a lot of genetic variants within that population, so as to generate phenotypic variation, those desirable characteristics that we're looking for. Also, an interesting thing to look at would be the hybridization or the crossbreeding between species within the Pleurotus genus, the oyster mushroom being Pleurotus austriatus. So the breeding of two different species with desirable traits in the hope of generating a genetically superior strain. And there have been studies that have shown that some of the species within the Pleurotus genus do successfully crossbreed. So that will also be an interesting way to look forward in generating an optimal strain.
Yeah, exciting.
Bioinoculants is also another thing I thought about, the interaction between fungi and their microbiome. And it's been well noted that some species of fungi do form mutualistic relationships with bacteria in nature, in their natural environments. And this has been observed in several instances where the fungal growth is positively affected by the presence of bacteria. There's been several instances which you can see certain examples. Some bacteria produce vitamin B1 for certain fungal species, and in turn, the fungus provides a vector for transportation of the bacteria. The bacteria use the growing mycelium to transport themselves through the soil, and so that they form a mutualistic relationship where the vitamin B1 benefits growth. Vitamin B12, which the oyster mushroom cannot synthesize itself, also benefits growth and is bacterially produced. Indole 3-acetic acid has been proven to stimulate oyster mushroom mycelium growth. And lastly, bacterial secreted cellulase. Cellulase is an enzyme that degrades cellulose, which is one of the components of hemp. And so while hemp does secrete its own cellulase, the supplementation of bacteria that also produces cellulase will make increase the bioavailability of carbon and improve growth characteristics, which is obviously what we're looking for. And these are just a few examples among many.
Yeah.
Additionally, another example of a study was performed by Young and colleagues on the almond mushroom, Agaricus subridescens. The study aimed to characterize and isolate bacteria found in the soil microbiome of the almond mushroom. And one of the bacteria that they found was Microbacterium humi. And they cultured the almond mushroom and they cultivated it with this bacteria. And what they found was a reduced time to harvest, as well as an increase in yield of up to 250%, which is obviously very impressive. And although these two characteristics, reduced time to harvest and mushroom yield, are not characteristics that we're interested in
Directly.
interested in improving or optimizing, it's very impressive to see the effect that bacteria and bioinoculants can have on the productivity of fungal strains. Another thing to look at, and what we've spoken about before, is the need for sterilization of substrates, obviously to kill off any bacteria or fungi that might compete for the growth of the oyster mushroom in substrate. And one thing that could be interesting would be the introduction of antimicrobial genes to our fungal strains, so as to give them their own means for sterilization and combating against opportunistic bacteria and fungi that would compete for nutrients. And obviously this would lead to a decrease in the chances of contamination and also less intensive sterilization of the substrate on our part, so that we might save resources in the sterilization of the substrate. And now moving on to something a bit more fun with the pigmentation. Moving away from functional properties and more towards aesthetic, there have been attempts in the past to bring about pigmentation in mycelium composite materials using food dye. Unfortunately, the mycelium uses the dye as a nutrient source, thus rendering this method effective. One potential way to circumnavigate this would be through the introduction of pigmentation genes derived from other plants or bacteria into the fungi, creating fungal strains with different colors.
Cool. Apologies.
You can see in the above diagram here, there's a vast spectrum of colors that have been characterized from bacteria, pretty much encompassing the full spectrum of colors. Once we have optimized the production process of the mycelium composite materials, we could create different pigmented strains to satisfy the potential demand for different aesthetics.
Cool. Now on to something that mushrooms are quite well known for, and we hope to utilize in the future. Back from the fun stuff to the functional stuff of mushrooms.
So bioremediation is something that, as Luke said, mushrooms are well known for. They can grow on a wide variety of substrates as a result of their wide array of extracellular enzymes that they can secrete. And utilize multiple substrates as a nutrient source. One thing that would be interesting would be the incorporation of food wastes into our substrate, kind of as a supplement for the growth of the bacteria. One such example is coffee grounds, which is high in nitrogen, or orange peels, which are high in multiple vitamin Bs, which, as I mentioned earlier, have been shown to benefit mycelium growth as a result of the mutualistic relationships between bacteria and fungi. Furthermore, some fungal enzymes have shown the ability to degrade certain plastics. While obviously the addition of such compounds into composite materials is not recommended due to the potential for residual plastic left over in the composite materials, it's certainly something interesting to look into, and certainly opens up the door to future opportunities and different technologies to utilize using mycelium and mushrooms.
So instead of just using the hemp agri-waste, which is another waste stream that we're going to use to create the hemp packaging products, now there's the potential to even intercept food waste and other harmful waste that generally lands up in landfill. Cool. So there's so many reasons around us why we should be doing this. This is not just science for fun. This is science for the future. It's science for sustainability, and something we believe in a lot at M2Bio, and we should all play our part in improving the circular economy.
Yeah, it's clearly evident that plastics and petrochemicals, which are obviously often used in packaging materials that we use today, which aren't biodegradable, which aren't sustainable, it's resulted in a lot of environmental harm, pollution, and even human and animal health, which is obviously concerning, which is shining a light on the potential need for an alternative.
Yeah, and there's definitely a tide turning on the traditional methods of creating packaging. We all become more and more aware of the disastrous effects they have on the environment and our health. Some more interesting.
And so obviously with the world's population ever increasing, the demand for packaging materials will increase and follow suit. And with that has been an increase in demand for sustainable packaging materials. One study by the Trivium Packaging's 2021 Global Buying Green Report showed that 83% of young consumers are willing to pay more for sustainable packaging, and 70% of all consumers are also willing to pay more for sustainable packaging, which is certainly eye-opening numbers. And on top of that, 2/3 of consumers consider it important that the products they buy use recyclable packaging. And this figure has not changed throughout the pandemic, which is also very interesting and shines a light on the fact that people are opening their eyes and becoming more open to this new alternative and being more environmentally conscious and wanting to make a difference.
Definitely. And if you look at those stats in contrast, the size of the packaging industry and 83% of young consumers interested in paying more for sustainable packaging, there's a huge market share to capitalize on there with the sustainable packaging that we're looking at. Great. Thank you so much for attending, and thank you, Aidan, for all that information. If there's any questions, please feel free to ask.